Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about laboratory calculations about serial dilutions. And here's the problem. 4 tubes were placed in a rack. To all 4 tubes 9 milliliters of phage buffer was added. 1 milliliter of a medium titer phage lysate was added to the first tube and mixed well. 1 milliliter of the first tube is transferred to the second tube and again mixed. The transfer of 1 milliliter from one tube to each subsequent tube continues until 1 milliliter is discarded from the last tube. What is the dilution of the phage lysate in the fourth tube? Let's say this is tube number 1, 2, 3, and 4. Now imagine that this is going to be a buffer which 9 milliliters plus 1 milliliter of the phage lysate. So 1 milliliter. So total we have 10 milliliters. Then we add 1 milliliter of the solution from the test tube number 1 to the test tube number 2. Again, we also have 10 milliliters here. Mix well and take 1 milliliter and add to the test tube number 3. Again, mix well, take 1 milliliter from the test tube 3 and add to the test tube 4. And again, we have here 10 milliliters. So what is the dilution we are going to get here? If you think that we have a ratio here of the solid to solvent as 1 to 9, so we just have to multiply 1/9 4 times, and this is going to be how we are going to get a right answer. Actually, this is not the correct variant how to solve this type of problems. Actually, what we have to do, we have to divide solid by solid plus solvent. And we are going to get 1/10. And this is what we are going to get every time we transfer 1 milliliter in each consecutive test tube. So what actually we have to do, we just have to multiply 1/10 4 times. So 2, 3, and 4. In numerator we have solid, in denominator we have solid plus solvent, so solution. The volume of the solid we have to divide by the volume of the solution. And the answer is going to be 1/10,000. So this is going to be our answer. This is going to be a dilution that we are going to get in the test tube number 4. And this is all for today. Subscribe and see you in the next video. Goodbye.